Welcome back, everyone, to another deep dive on pharmacy and medical focus. I'm so thrilled you're hanging out with us today. We're unlocking what I like to call solubility secrets, the absolutely fascinating science behind pharmaceutical solutions. You know, a bottle of liquid medicine might look super simple from the outside, but actually turning a solid chemical drug into a stable, palatable, and effective liquid dose, that takes some literal genius in pharmaceutical engineering. Okay, let's dive right in. It's a high-stakes journey of transforming solid drugs into life-saving liquid solutions.
Here's our roadmap for today's explainer. First, the anatomy of a solution. Then, solvents and excipients, followed by routes of administration. We'll look at formulation trade-offs, and finally, enhancing solubility. Stick around for that last part, because we're going to build up to some brilliant biochemical hacks that are just mind-blowing.
All right, part 1, anatomy of a solution. Before we get into the really fancy stuff, we have to understand the core problem. How do we get a solid drug perfectly and evenly dispersed into a liquid?
So, by definition, a pharmaceutical solution isn't just your average mixture. It's a homogeneous molecular mixture of a solute, which is usually our drug, dissolved in a solvent. And that gives us an optically clear liquid. The big takeaway here is that because the drug is completely dispersed as individual molecules or ions, it's immediately available for your body to absorb. There's no waiting around for a pill to break apart in your stomach. It is ready to go to work right away.
Now, water is obviously the most common solvent we use, but that actually raises a pretty provocative question. Why can't we just use regular tap water to make liquid medicine? I mean, it's cheap, it's everywhere, and it's totally safe to drink, right? Well, potable tap water actually contains dissolved substances, minerals, and trace elements. And while those are totally harmless to drink, they can completely ruin a highly sensitive drug's chemical stability and solubility. Because of those risks, pharmacopoeias have really strict grades of water. Purified water is used for basic, non-sterile medicines. But look at the requirements for parenteral solutions, like IVs. They require water for injections, which absolutely must be strictly pyrogen-free. Pyrogens are water-soluble, fever-producing compounds, so if you used standard water for an IV, you could accidentally induce a massive, dangerous fever in a patient who's already vulnerable.
Moving on to part 2, solvents and crucial excipients. Let's move beyond just water and meet the alternative solvents and the supporting cast that makes up a pharmaceutical solution. We basically have two main systems here. Aqueous solvents are water-based. They have low toxicity and they're cheap. But here's the thing, not all drugs like water. So, the crucial point is that when a drug is unstable in water or just stubbornly refuses to dissolve in it, we have to turn to non-aqueous solvents, like specific oils or alcohols.
And just look at the sheer variety of real-world alternatives we can use. Take fixed vegetable oils, for instance. We're talking about highly purified olive oil, sesame oil, or corn oil. Historically, and honestly even today, these are used for intramuscular injections. You might be wondering why. Well, injecting a drug dissolved in oil creates a sort of depot in the muscle. That depot slowly releases the drug over time, giving the patient sustained absorption. Pretty clever, right?
Alongside the solvent, we have the excipients. Think of them as the supporting crew. These are substances added to enhance stability, bioavailability, and just overall patient acceptability. You've got sweeteners for taste, antioxidants to stop chemical degradation, and preservatives for microbial safety. But let me tell you, the stakes are incredibly high here. For example, using the preservative benzalkonium chloride in an oral drop or an eye drop, perfectly fine. But if you formulate a pulmonary nebulizer solution with it, it causes dangerous bronchoconstriction. The patient's airways will literally tighten up. Context is absolutely everything.
Formulators rely on this exact toolkit of excipients to keep the drug alive on the shelf. They'll use chelating agents like dis-sodium edetate to essentially lock up heavy metals that might catalyze oxidation. Sometimes chemical degradation is such a massive threat that formulators will literally purge the liquid with nitrogen gas. They create a nitrogen headspace right there in the bottle just to forcefully exclude oxygen and stop the drug from degrading. It's intense.
Okay, part 3, routes of administration. We have our perfectly crafted liquid, but how is it actually deployed into the body? Let's take a look. The journey of a liquid dose depends entirely on its destination. The oral route is relatively forgiving. It mostly requires palatability, and it can tolerate a surprisingly wide pH range, from about 2 to 9. But move over to the ocular route like eye drops, and suddenly you need strict sterility and isotonicity just to match natural human tears. Go one step further to the parenteral or IV route, and the requirements for sterility and being pyrogen-free are absolute. No exceptions.
Let's see how this translates into real physiological stakes. For parenteral requirements, intravenous solutions absolutely must be purely aqueous. If you accidentally administer an IV solution that contains oil droplets, those droplets can travel straight through the bloodstream, occlude the pulmonary microcirculation in the lungs, and cause a fatal blockage. Furthermore, if an injected solution doesn't have an appropriate pH, it can cause excruciating pain, phlebitis, and even literal tissue necrosis, which means the surrounding tissue actually dies.
Which brings us to part 4, the formulation trade-offs. Because of these massive physiological consequences, pharmaceutical scientists are locked in a constant battle between creating an effective medicine and keeping it stable.
But when they win that battle, the benefits are literally life-saving. Solutions offer rapid drug action because the drug is already molecularly dispersed. Think about an adrenaline injection during anaphylactic shock. It works instantly because it doesn't need time to dissolve first. Solutions also give us perfect dose uniformity, and they're vastly easier for young kids and the elderly to swallow compared to those giant, chalky capsules. But, the drawbacks are pretty severe. Water is kind of the enemy of stability. Being in a molecular liquid state makes drugs incredibly vulnerable to heat, light, and chemical reactions like hydrolysis and oxidation. On top of that, liquids are heavy, they're bulky, and they're wildly expensive to manufacture and transport compared to dry powders. And honestly, most frustratingly, some of our most amazing drug compounds are just incredibly poorly soluble in water.
And that brings us to part 5, secrets to enhancing solubility. This is the climax of our deep dive. What happens when a miracle drug stubbornly refuses to dissolve in water?
Well, we start with 2 primary biochemical hacks. First up, pH adjustment. Most drugs are weak acids or weak bases. By carefully tweaking the pH of the water, we can actually force the drug to ionize. And guess what? Ions love water, which dramatically boosts solubility. Second, we use cosolvents like ethanol, glycerol, or propylene glycol. Water is very polar, and nonpolar drugs hate it. But by blending in a slightly less polar cosolvent, we lower the overall polarity of the water, essentially tricking the nonpolar drug into dissolving.
But what if even that isn't enough? Enter the ultimate molecular Trojan horse. A cyclodextrin molecule beautifully illustrates this concept. Structurally, it's like a hollow, truncated cone. The exterior rims are covered in hydroxyl groups, making the outside totally hydrophilic. Water absolutely loves it. But the hidden interior cavity? That's nonpolar and hydrophobic. So formulators use cyclodextrins to basically swallow up stubborn nonpolar drug molecules, shielding them right inside this water-soluble cone. The body just sees a happy water-soluble molecule while the drug is safely smuggled inside. How cool is that?
Finally, we have Micelles solubilization. Scientists use surfactants, which are molecules with a water-loving head and a fat-loving tail. When you add enough of these to water, reaching what's called the critical micellar concentration, they spontaneously self-assemble into microscopic spheres called micelles. The fat-loving tails point inwards, creating a hydrophobic core. We can then trap highly insoluble drugs right inside that core. This incredible technique is exactly how we're able to take notoriously stubborn fat-soluble vitamins, like vitamins A, D, E, and K, and successfully formulate them into aqueous liquid injections.
It really makes you wonder, right? If we can use pH, cosolvents, cyclodextrins, and micelles to molecularly trick water into dissolving almost anything, what formerly impossible drugs might become the liquid cures of tomorrow? The science of pharmaceutics is constantly pushing the boundaries of what is possible. If you find these solubility secrets helpful and want to keep exploring the fascinating world behind your medications, please hit that like button and subscribe to Pharmacy and Medical Focus for more deep dives into the science of medicine. Also, just a quick reminder, the content provided in this explainer is strictly for educational and informational purposes. It doesn't constitute and shouldn't be used as a substitute for professional medical advice, diagnosis, or treatment. Always seek the advice of your physician, pharmacist, or other qualified health provider with any questions you might have regarding a medical condition or medication. Thanks so much for joining me today. Keep asking great questions, and I'll see you in the next one.